We are aware that there is a wide variety of RNA viruses in nature, particularly the recent rampant novel coronavirus, which is also an RNA virus. Compared to double-stranded DNA, single-stranded RNA is less stable and more prone to mutations during replication, leading to a greater diversity of RNA viruses, making them challenging to prevent. In response to this situation, researchers have been continuously exploring solutions to mitigate the threats posed by RNA viruses to human society. Today, we will introduce a novel RNA virus detection approach based on transcription amplification and the CRISPR-Cas13 system. This approach offers a detection sensitivity of up to 82 copies of RNA targets and can accurately differentiate critical mutations. The basic concept employed in this study is as follows. Firstly, targeting critical regions of the RNA virus. Two complementary strands, pre-substrate A and pre-substrate B, are designed. When the target RNA is present, pre-substrate A and pre-substrate B can be ligated by the action of T4 RNA ligase 2, forming a complete pre-substrate chain. This complete chain serves as a template for T7 transcription and undergoes in vitro transcription in the presence of the T7 promoter and T7 RNA polymerase system, resulting in the generation of an RNA substrate chain. Subsequently, the CRISPR-Cas13a system, guided by specific CRRNA, selectively recognizes the RNA substrate chain and activates reverse cleavage activity. In this study, a signal reading step introduces the RNA aptamer fluorescent probe DFHBI1T. DFHBI1T produces high fluorescence when bound to the broccoli RNA aptamer, but remains non-fluorescent otherwise. Utilizing the reverse cleavage activity of Cas13a, which is activated specifically by the target, the broccoli RNA aptamer is cleaved, leading to a significant decrease in fluorescence, thereby indicating the presence of the target RNA. To validate the feasibility of this detection method, the fluorescence values of the system were tested under different component conditions. In the presence of only DFHBI1T, the fluorescence values were extremely low. However, the addition of the broccoli RNA aptamer resulted in a 430-fold increase in fluorescence, which is advantageous for achieving a high signal-to-background ratio in the entire signal detection system. The addition of Cas13a CRRNA did not cause any significant changes in fluorescence. However, the presence of the substrate RNA led to a significant reduction in fluorescence intensity. The results indicate that the substrate RNA activates the reverse cleavage activity of Cas13a, leading to substantial digestion of the RNA aptamer and a significant decrease in fluorescence. To further verify whether the ligation reaction itself can trigger the detection signal, researchers conducted a test in the absence of T7 RNA polymerase. In the presence of the target RNA, the absence of T7 RNA polymerase resulted in no significant change in fluorescence values. This suggests that the substantial cleavage of the broccoli RNA aptamer is the result of initiating transcription amplification. This process was further confirmed through gel electrophoresis analysis. Lane 3 showed a lagging band, indicating the binding of target RNA segments with two precursor segments. The further addition of T7 RNA polymerase promoted the production of a substantial amount of transcription substrate RNA. The results indicate that the signal triggering in this detection system is primarily initiated by transcribed RNA. Apart from RNase virus typing, the ability to analyze RNA virus mutants is also crucial, as it allows for precise estimation of epidemiological trends and pathogenic risks. To evaluate the detection scheme's capability to identify RNA mutants, Researchers took the example of novel coronavirus and detected the critical D614 gene mutation. They designed a series of pre-substrate probes targeting the D614 gene mutation and sequenced both wild-type and mutant pseudoviruses of SARS-CoV-2, confirming the presence of the D614 gene mutation. The results indicated that the D614 gene mutation can be significantly distinguished through RNA detection, whether it is anchored in the pre-substrate A probe or the pre-substrate B probe. Researchers further assessed the recognition rate and found that mutation positions near the connection site of the pre-substrate probe resulted in better recognition rates. In order to improve the specificity of the detection, researchers also designed a series of CRRNAs with different recognition sequences to screen for target sites. The results show that the optimization of Cas13a CRRNA recognition significantly improved the recognition ability of the entire detection system for D614 gene mutations. In addition, the researchers estimated the ability of this detection method to distinguish D614 gene mutations in the presence of wild virus RNA. Compared with the D614 gene mutated viral RNA, the increase in wild viral RNA concentration only slightly reduced the fluorescence of the detection method. Even the number of wild virus RNAs is 50 times that of D614G mutated virus RNAs, and the fluorescence reduction is much smaller than the presence of D614G mutated virus RNAs. The results indicate that this method can even identify a small number of mutations in virus RNA samples. The dual recognition mechanism of connecting reaction and Cas13a CRRNA ensures the detection system's ability to distinguish single nucleotide mutations. So, in conclusion, in the featured presentation, researchers have developed a novel RNA virus detection method based on ligation, transcription, the CRISPR-Cas13a system, and RNA aptamer fluorescence. This approach allows for sensitive detection of target RNA without the need for reverse transcription and pre-amplification, and can distinguish mutants. The strategy is capable of specifically targeting a range of RNA viruses, including coronaviruses such as SARS-CoV-2, MERS and SARS, as well as influenza viruses like H1N1, H7N9, and H9N2. The dual recognition process involving ligation and Cas13a CRRNA ensures high specificity for distinguishing single nucleotide variations, making it a promising tool for on-site monitoring of RNA viruses and their risk mutants.